Good morning to all our viewers in the US. Good evening to our viewers in Europe, and a good day to all our viewers in other parts of the world. Thank you very much for taking the time today to join our webinar on the peptide Easy Clean technology conducted by Bachem in collaboration with Belintic. Before we begin, I would like to give a brief introduction. Bachem is a global CMO and industry leader in the synthesis of peptides and oligonucleotides. We celebrated our 50th birthday last week, and we're well known for setting industry standards in the manufacture of high-quality peptides for various markets, including pharmaceuticals, cosmetics, and diagnostics. We operate internationally, and our corporate headquarters are located in Bubendorf, Switzerland, just outside of Basel, and we have locations in Europe, the US, and Asia. And for those who are interested, we are listed, on the Swiss Stock Exchange, and more details about the company can be found on Bachem's website under Investor Relations. I'd like to take the opportunity to introduce our speaker for today, Dr. Robert Zitterbart, who is the co-founder and head of R&D at Belintic. Belintic is in Berlin, Germany. And at Belintic, Robert is focused on developing linker systems to purify and modify peptides. Robert has almost a decade of experience in the development of peptide purification technologies. After his diploma work in the group of Professor Peter Volhard at UC Berkeley, he earned his PhD at the Humboldt University in Berlin in the group of Oliver Seitz. During Robert's scientific career, he has successfully published many articles in peer-reviewed journals. And for those who are interested, his latest paper on the peptide Easy Clean technology was published in Chemical Science. I'd also like to take the opportunity to introduce myself. My name is Arjun Narayanan, and I am a technical sales specialist supporting the sales of research-grade peptides for Bachem Americas. And I just completed 2 years at Bachem last week. So it's time for our first poll. So the question I'd like to ask all our viewers is, if any of you work with modified peptides. You should see a list with 4 options. Please choose the option that is most relevant for you. All right, so the results are being displayed on the screen, and it's interesting. Most of you have said that yes, you do work with modified peptides, and some of you say that yes, some of these modifications do cause trouble. And for those of you who are experiencing problems with peptide modification, we're glad to have you here, and hopefully you find this webinar very useful. And we'll continue with our introduction. So Bachem, as I mentioned earlier, has just completed 50 years of business, and we do provide products for research, clinical development, and commercial application to not just pharmaceutical and biotechnology companies, but also to cosmetics, diagnostics, and academia. And at Bachem, we focus on what is known as a 360-degree business model, where we like to work with customers at all stages of the product or drug development process, right from initial screening, lead development, all the way through to commercial API production. We are located at various parts of the world globally. As mentioned earlier, our corporate headquarters are in Bubendorf, Switzerland, just outside of Basel. And in the US, we operate under the Bachem Americas umbrella. And the Bachem Americas operations are based out of Torrance, California, which is just south of Los Angeles. And we also have a secondary site in Vista, California, which is near San Diego. In addition, we also have a site in St. Helens, UK, which specializes in the development and manufacture of research-grade peptides, as well as a sales office in Tokyo, Japan, and a secondary site in Vienna, Switzerland, which specializes in the large-scale manufacture of small molecule APIs, as well as our FMOC amino acid building blocks. And now to introduce Bilintic. Bilintic is a German startup with strong expertise and innovative potential, and Bachem identified them, and we've been nurturing a sustainable partnership with them right from the beginning. Our joint focus is on the development of innovative technologies to explore new possibilities for purification and manufacture of peptide APIs in order to better serve our customers. And what Bachem brings in this partnership is our strong expertise and 50 years of experience in the development and large-scale manufacture of peptide APIs. And we're very proud to host Bilintic for our webinar today. And this is a continuation of our earlier webinars with Bilintic. And this webinar will potentially provide insights on how the peptide Easy Clean technology can be used to improve API properties and be used to not just purify peptides, but also modify peptides. And with that, I would like to turn it over to Robert. Before Robert begins though, I'd also like to mention that we do have a Q&A function, and with that, you should be able to see the Q&A button, at the bottom of the screen. Please use that to input any questions that any of you may have at any time. And at the end of the webinar, we will have time for questions, and we'll do our best to address as many of the questions that you may have. And with that, sorry, I will turn it over to Robert.
Thank you, Arjun, very much for your kind words and introduction. And thank you all of Bachem for having invited me here to present this talk on your webinar with the title Peptide Easy Clean is a highly efficient tool for peptide modification. But before I explain about the peptide modification, I will give you a short agenda, what I'm going to talk about. So first I will introduce Bilintic, and then I give you a detailed look on the PEG technology in general, also give high throughput purification examples. And then it's actually then about the peptide modification facilitated by PEG. And then some closing summary remarks with also, I will touch a little bit on scale-up considerations. So first, who is Bilintic? So as Arjun already introduced a little bit, we see ourselves as an independent chemistry for healthcare enterprise, and we try to enable powerful peptide drugs or peptide therapeutics against widespread diseases. We and our team, we have a strong background in peptide and organic chemistry, and how we want to improve these powerful peptide therapeutics in manufacturing. Therefore, we have developed our technology, and therefore we own an unrivaled peptide manufacturing technology that I will explain to you in a second. We think we can, with our technology, address the whole value chain of peptide therapeutic, of the whole value chain of the peptide therapeutic markets. So first, by starting in the discovery phase, where we can offer like high throughput solutions for peptide purification that is unmet currently, but by status quo technologies. Also in the development phase where you have less peptides and numbers, but higher scales, and maybe you want to have more complex peptides, for example, modified peptides. So with that, we can enable these peptide therapeutics through our first-in-class modification platform. Also in production scale, we think that with our technology, you are more flexible and it's more easy to scale up. I will show you also one example where we did a scale-up study. And also in commercialization phase, when an API candidate has been approved, we can also leverage productivity by increasing the productivity in a peptide manufacturing process with our technology. Okay, so the basic products we offer are kit products, as shown here. So we have high throughput kit products where you can use our technology in a plate format, or research kits where you have different sizes and you can purify peptides manually already in such cartridge filters here, or developer kits that are more bulk material kind of products. And what this means in detail, I will just give you an example. So in these high throughput kits, we offer two formats. One is 96 well format, where you can purify 96 peptides in parallel and get approximately 5 milligrams of each purified peptide. Of course, depends on the molecular weight of the peptide. Or you can use a 48 times 20 or 25 micromole scale. This will give you roughly 10 milligrams each of purified peptide. Or as I mentioned, the research kit comes also in 3 sizes, whether you purify in these cartridges, not in plates. So here you purify in plates, here in cartridges, whether 24 peptides, also 5 milligrams each, or 8 times 30 milligrams each, or 8 times 50 milligrams each. Always depends, as I said, on the size of the peptide, of course. And the developer kit gives you basically the possibility to purify an unlimited number of peptides, or you aim for scale up. And the biggest developer kit we offer has 100 grams of linker molecule, and this gives you roughly, again, depends on the peptide, but as a rule of thumb, 100-gram purified peptide. Also, we offer services. So currently, we also offer peptide library services, where we use our high throughput kit to purify peptides that we have synthesized ourselves as of basically feasibility studies to show basically the quality of the peptides. They perform very well in assays. Also, we offer like co-manufacturing as a service. If you have problems with a particular peptide, we offer to give you all our knowledge and with our technology to leverage this particular peptide. Or we also offer like co-development services, where you have a particular peptide, but you want to, or several peptides, and you want to improve of the pharmacokinetics. We can run our modification platform on that, so modify each position that makes sense with our technology, and hopefully improving the pharmacokinetics of the peptide. So, but again, to our technology in detail. So first of all, this is a general scheme. I will also give a deep dive in the chemistry later. But here is an overview. So in this scheme, it shows the amino acids as purple spheres. So as you know, in solid phase peptide synthesis, the amino acids are coupled after each other. To use our technology effectively, we recommend to use capping with acetic anhydride or other capping means. And this gives you the opportunity to couple the PEG linker as the last amino acid or last residue on the full-length peptide. Therefore, your desired peptide is tagged by the linker. Then you can do your normal TFA cleavage, giving you the crude product mixture. You can dissolve it and then put it to the modified agarose beads that we are using on a standard basis, or other materials we have investigated. This allows the selective catch of the desired peptide, and the synthetic dirt can be washed out. And then a releases the desired peptide from the purification media, giving you the purified peptide. So since this webinar is now also about modified peptides, one can use also this at this stage here, not to just release the peptide, but you can also use this already like pre-purified peptide to do modifications on the PEG phase. Therefore, you can conjugate it with, for example, lipids or PEG, or even biomolecules on that. And then after release, you have a conjugated purified peptide, or you can perform cyclizations on the PEG phase. For example, thioether stapling, as I will show you on some slides, or disulfide formation. And then also, again, after release, you have your purified cyclized peptide. So this is all possible by the PEG linker. And as Arjun has mentioned earlier, we have just recently, also together with Bachem, published a chemical science paper that gives you all the details about the chemistry. But so all the magic has been done by this PEG linker here that consists of 3 key components. Here on the left side, you have a protected amino oxy group that enables a catch, or this is a catch tag for immobilization of the desired peptide via oxim ligation. And here on the middle in green, you have a cleavable unit. This is an aryl A site. And on the right side, you have a leaving group that enables the attachment of the desired peptide to the linker molecule. And here it's basically the same scheme as I showed before, but in more chemical details. So to make this clear to everybody, let's assume we want to synthesize a sequence peptide, right, by these amino acids. And as you all know, in the course of SPPS, you build up truncations or deletions even. So meaning that there are sequences where the one amino acid coupling was not quantitative. So in this particular example, let's assume like there's one troublesome junction, the coupling of the proline P here on the T on treonine. Let's say it just works like 50%, leaving the rest of the treonine sequence tied with a free N-terminus. So when you do the capping after the proline coupling, the proline is still FMOC protected. After capping, you have capped this N-terminus, and then it starts all over again. You would deprotect FMOC and couple the next amino acid, and so far. So this enables you then to have the peptide of interest with the sequence peptide with a free N-terminus, and then you can couple the PEG linker on it. And the truncations are not touched by the PEG linker. And then the peptide is still protected, so is the linker, and still attached to the SPPS resin. TFA cleavage liberates the peptide, also releasing the amino oxy group here. Then you can dissolve this mixture and incubate it with the activated filter material. This is aldehyde modified, enabling a robust and fast oxime ligation, giving a covalent bond of your desired peptide to the purification resin. And then you can wash away all unwanted synthetic dirt and truncations. And then a safety release liberates the peptide, and you have your purified peptide sequence. And what the safety release is, I will show you on the next slide. This is also something we describe in very much detail in the paper. So this linker electronics allow it that you can reduce the A site to an aniline here, to the amine, by, for example, usage of DTT. And but the linker is still stable, therefore we call it safety release. So it's just primed for release. This enables the washout of the reducing agent, and only upon addition of acid, the carbamate is protonated, enabling or giving the trigger for 1,6 elimination. This is releasing then eventually the peptide. The rest of the linker stays on the resin. What is also interesting is that depending on what acid you use for this acid trigger, you can generate the desired salt form. So you don't have to use TFA, but in fact, we mostly use TFA. But you can use HCl or acetic acid as well. What is this technology good for? So what is very useful is it's a completely orthogonal purification method orthogonal to HPLC purification. So impurities are removed chemoselectively, and it's also more a one-size-fits-all solution than HPLC is. So there's no method development or anything needed. But the orthogonality is clearly shown here on this particular peptide that we have purified together with Bachem UK, where at first glance, the crude chromatogram without linker looks quite clean, but on close inspection of the EasyMS mass under this peak, several truncations are revealed. This is a quite hydrophilic peptide, so it has many arginines and lysines, for example. But after the PEG process, so all these underlying impurities could be easily removed. And this was not feasible by HPLC. So Bachem UK had to use a had to use a two-dimensional purification by using two acidic modifiers. Okay, but to give you more examples on adjusted purification, I will just rush through this. So again, a new antigen, or not again, but we have shown parts of this in the first webinar. But you can also read it in our papers, and the sequences of this neoantigen personalized medicine set is derived from this paper here from BioNTech actually. And so, and in this particular case, we wanted to purify 20 peptides as fast as possible using our technology. So, and I wanted to show you this again to show you how the technology actually looks like. So it starts with a SPPS, where the linker can be coupled as well. But in this case, there has been performed a tri-cleavage before linker coupling. So then it looks, this particular peptide 1 looks like this. After unoptimized synthesis, because in these neoantigen sets, you don't have time for optimization, obviously. And then after linker coupling, the desired peptide always shifts a little bit to longer retention times, here marked by this asterisk. And then after incubation of the crude mixture with the agarose material, you see all the linker tagged peptide is gone. And you can also already analyze the supernatant, and you see that most of the dirt is also not binding to the agarose. But washing is nevertheless recommended to get a higher purity, and then the release then in this case looked like this, so gave the peptide in 91% purity. And so overall, here's a box diagram of the purities. So the crude purities were quite widespread, and we were able with one PEG run to increase the mean purity from 53% to 89%. And for example, for this particular peptide, this was most impressive because this was actually an impurity that was in intensity higher than the desired product, was co-eluting, and this was again no problem to remove this. And the whole set was purified in a single day in 3 active hours. If you want to go for even higher throughput, you can use our high throughput kits and plate format. So it looks quite similar, just that the synthesis in some plates and the purifications in plates. We recommend to use these vacuum manifolds we also provide. This gives you the final peptides, whether again in 96 well format or in 48 well format, and then you have your lyophilized peptides right away. And we have done this now many times. It works quite well. Here's some data I'm allowed to show. When we started with this, we purified a 96 peptide library for Professor Stephen Cobb from University of Durham, UK. And in this case, we could improve the mean purity from 70% to 88%. Oh no, the mean was 67. So this is the median purity, the 70. So the mean purity from 67% to 87% could have been even higher, because like from these 96 peptides, 72 peptides were prone to aspartamide formation, and this is unfortunately an impurity we can not remove, but we can or one can do synthetic measures to avoid this impurity. So now to the main topic of the talk. So how the Peptide Easy Clean technology can facilitate modification of peptides. So maybe a word on the modification. What we mean by modification is just basically an alteration from the normal 20 proteinogenic amino acids, and usually also like a modification, a side chain of a amino acid. So there are basically two different approaches how to use or how you can benefit from the PEG technology when you handle modified peptides. So the basic approach is A. Where you introduce a modification before you do the PEG run by using, for example, a modified building block. And then, as I explained to you before, you add the linker and then you just purify your desired peptide. And the second approach, B, is, as I showed you before, that you do the modification on the PEG solid support. But first I will talk about the A way. So the modification before PEG. So what is the benefit here? The benefit is clearly that PEG, as for hydrophobic peptides, offers more freedom in dissolution of difficult to be handled or purified peptides. If you have very hydrophobic peptides, PEG also helps because you can use any solvents also in larger amounts to dissolve them, and still the oxime ligation is very robust and works. And but if you have lipidated peptides, they also become, by the modification, very hydrophobic. Therefore, it also helps that you can dissolve them more easily. So, for example, you can dissolve impure DMSO, and then what we usually do is add 10% of buffer, pH 4. Actually, what's missing here is that we also have in this particular buffer gluidinium chloride to prevent hydrogel formation or aggregation, and this works for almost all peptides. First dissolution DMSO and then the addition of gluidinium chloride. But for very hydrophobic peptides also, especially when they are lipidated, you can also use HFIP, so hexafluoroisopropanol, that dissolve very well, especially hydrophobic peptides. And what's also feasible, not so much with the agarose, but with other material we have, you can dissolve in like TFA and immobilize directly out of TFA. So this is also a big bonus. And yeah, again, the biggest benefit here is that you can purify or handle difficult to purify hydrophobic peptides, especially where the modifications, especially lipidation, makes them so hard to handle. And we have showcased this on example of liraglutide that is also known to be not easily handleable because it tends to aggregate. It's very hydrophobic because of the lipid side chain. It tends to form fibrils or anything. So so in this modification A part, so modification before PEG, we used this already modified lysine building block where the SPARM glue was already attached to the lysine. This unfortunately makes the rest of the SPPS not so straightforward. Therefore, we also introduced like 3 pseudoprolines to still have a reasonable crude purity. And then we did the linker coupling on this TFA cleavage and the PEG purification. And we did this in 0.02 millimole, 0.2 millimole, and 2 millimole. In this first two examples, we used our basic products, the kit, what also sold on the kit, so in cartridges. And for the 2 millimole, we just used normal organic glassware, and it can be easily done. So this is how it looked like. So here at the top, you see the crude purities before linker coupling. So of batch 1 and batch 2, so the 20 micromole and 200 micromole purification attempt was used from the same batch, so obviously the crude purity is the same, and the purified purity was also in the same range, giving exactly or almost exactly tenfold of the amount that we got in the 20 micromole scale. We got then in the 200 micromole scale, and what was really we were really happy when we then transferred it to 2 millimole. This was a different synthesis batch, so slightly less purity, and we also had slightly less purity in the end. But what made us very happy is that the amounts really scaled linearly from 20 micromole to 2 millimole. So now I want to show you the modification by BiPAC, then the SPI approach. So the benefits are that you can use a highly chemioselective purification method. Also, when you have then immobilized your desired peptide on the agarose or on the PEG phase, it hampers the multimerization because you have a pseudo high dilution like at SPPS. And the N-terminus is intrinsically protected by PEG, so you don't have to use an extra protecting group. And you are not restricted in any solvents, and one point is missing here. You're also not restricted in equivalence, because you can just wash out the excess of equivalents or reagents because you have a solid support. Where can it help? For example, in PEGylation, also in cyclization, disulfide formation or stapling, where we'll show examples of biotinylation, lipidation, you name it. And again, I will show an example of the liraglutide, but one more time to the chemistry. So in this particular case, imagine we have not the sequence peptide, but pegtide with a K here, and we want to modify the lysine. So again, we modify it with a linker and have just one lysine free. By the way, you can optionally, you can pre-purify this by HPLC. That has the benefit that you can easily get high purities at this stage, because when you have lipidated your peptide, maybe not so much with one lipid, but maybe if you have multiple lipids, then purification can get very troublesome, and then this is an option. But independent if you do an HPLC or not, how it works then is you can immobilize again on the PEG support. Then you have the N-terminus is blocked by the linker, the amine is free, and then you can do acylation reactions by lipids or PEG, for example, and then you can release with acid to get your modified and purified peptide. In case you have done an HPLC purification and these steps, they usually don't introduce side reactions. If this is the case and you have purified your non-lipidated peptide, for example, to high purities, and the process maintains this purity, you can release already with the desired acid you may want to have in your final product. So this can be of big benefit. In this, so I will show you this, and again, an example of liraglutide. In contrast to the first example, we just linearly synthesized the liraglutide by normal building blocks without any pseudoproline, because when you use just the standard lysine building block, lysine BOC or lysine tritool, then the SPPS works well. And then we have linker modified it, immobilized it on our support, and then lipidated with the palm glue building block, and then released by TFA. And this also removed the tert-butyl protecting group on the of the palm glue building block, giving the purified liraglutide. How this looked like in chromatograms is shown here on the right side. So the crude purity without PEG linker was 55%. Then you see with the PEG linker, it shifts a little bit to the right side. If you immobilize this, you see quite some synthetic dirt in the supernatant. And then these modification steps are performed on on the support, and then you can release your peptide. In this case, we got a purity of 80%. We have not optimized this, so we got like a 2% of unmodified peptide. But by the way, I did the same thing actually on SPPS, and also on SPPS, the lipidation with palm glue is not so easy. So actually, we got better conversions on agarose than on SPPS polystyrene resin. But we also know from other projects we are not allowed to talk about that you can easily achieve 100% conversion. So then we would be at 82% purity, as in the previous example, actually. What you can also do is disulfide formation. Again, if you have a peptide with 2 cysteines, so septic, or how to pronounce it, then you have 2 free tiols here, and this can be modified. But in this case, if you want to have disulfides, you better reduce the linker before you oxidize, because otherwise it will maybe, depending on the conditions, also reduce a newly formed disulfide bond. Therefore, we recommend to reduce the linker. And as I said before, you have the safety release, so it's still stable. And then you can oxidize it and then release the disulfide peptide with acid. And here are 2 examples. So first, the vasopressin, quite small peptide, but a good starting point. We tested several conditions, standard conditions: iodine, DMSO, pure DMSO, PBS buffer, and chlorosuccinimide, and also this platinum complex, also referred as Rabenstein reagent. And all oxidants worked well. Only DMSO didn't give like quantitative results always, depending on the peptide. We also tested other peptides. NCS and Rabenstein worked best, but sometimes showed some over-oxidation. As you see here on the right side, it seems to be some over-oxidation. Another example is here shown on solnatide. It's a peptide that's used for treatment of COVID-19 patients that have severe respiratory failure. The API is in phase 2 currently, and has been approved for compassionate use in Austria. So it's currently used to treat COVID patients, and it's a disulfide peptide. And so we tested it as well without any optimization. So we just synthesized it and had a crude purity of 65%, and did the oxidation with Rabenstein reagent and PBS for 60 minutes, and got a final purity of 80% that could be easily polished by HPLC and 100% conversion to the disulfide. What one can additionally do is, for example, if you'll have like 3 cysteines in your sequence, you can also perform like stapling reactions where you add a scaffold here that reacts with the cysteines and then gives you this stapled bicyclic peptides. In this example, we have also done this on research peptides, where we have used like the standard benzylbromide scaffolds, TBMB in this case, and have shown that it works also well, that you can incubate those peptides with the scaffold on our PEG phase, and you can slightly improve the purity, and you have a good conversion rate towards a desired peptide. And in these 2 cases, the purity was increased by 10% and always 100% conversion. So this brings me almost to the end of my talk. So also some large scale considerations. So the recovery we get are, if you don't do any improvement, are usually between 40 and 80%, can be improved if you look on a particular peptide. But the agarose and also other material we have in stock has a high loading capacity of 10 to 20%. So this, I want to emphasize this here because usually, since our technology is mostly compared with HPLC, it's always a little bit hard for potential customers to grasp how this really looks like. But it's more comparable to SPPS really, because also the loading capacity is more like SPPS. So what we could already show is that you can perform the PEG purification and modification in SPPS reactors. We have not shown it in such a big reactor, but it's conceivable that it can be also transferred in such big reactors. And this would basically mean that in a 1,000 liter reactor, you can basically purify or modify up to 40 kilogram of product per day, maybe even more. This would have to be tested, of course. And what I have not emphasized yet is so first of all, you have less solvent handling than compared to HPLC, but also you save lots of solvents. So in research scale, we save like 75% or more of solvents. And I think also in scale up, if you like optimize it for a certain peptide, you can save, I would say, even more percentage, but this would have to be tested, of course. And the PEG linker and the agarose material can be supplied for GMP compliant processes in kilogram to ton scale. So let me summarize. So PEG enables a high throughput peptide manufacturing during early development stages. It serves as a tool for peptide modification during the lead optimization stage, for example. So, and there are two different routes. So there's a route A, where PEG enables the purification of SPPS modified peptides. This is beneficial because you have a higher flexibility in the solution of your complex peptides. And we have shown a linear scale up from 24 milligram to 2.4 gram. But you can also use it to modify peptides. So this has been shown, for example, by the lipidation or towards liraglutide, or by oxidation towards disulfide peptides showcased on vasopressin and solnatide. And also thioester stapling is feasible to gain bicyclic research peptides. Therefore, PEG is an attractive orthogonal method for, and also not only in research scale, but also for large-scale purification of specific modified peptides. Thank you very much for your attention, and I hope to answer your questions soon.
All right. Thank you very much, Robert, for that extremely interesting and useful presentation. Now, we have one final poll question for our viewers upon viewing this complete webinar. Do you think the PEC technology could help you? You should see a window with 4 answers. Please select the answer that that you feel is relevant now. And it looks like most of you are indeed interested in the PEC technology, and I'm very glad, Robert and myself are very glad to see that. That's great news, and that means that this webinar was indeed, very useful for all of you. I'm glad to see that. And now we come to the Q&A of the webinar, and in the time we have left, we will do our best to answer all of your questions. And I'd like to thank our viewers for all of their attention and for submitting many, many, many interesting questions. Some of these questions were indeed answered by Robert during the course of the presentation, but we'll answer some of the outstanding questions right now. The first question that that I'll ask Robert is, how effectively does PEC purify amyloid peptides, which are prone to aggregation? And do you have any data that has shown that PEC is effective for the purification of amyloid-type peptides?
Yes, we have not yet purified the whole amyloid beta sequences of the 42 mer. We have not yet purified, but we have purified like 20 mers of the sequence, and also like the one that is very aggregating. I'm not sure if it's like the C-terminal end or an N-terminal end. But in terms of aggregation, this was no problem. You have to make sure that you have guanidinium chloride or urea inside in the dissolution, because otherwise you will get some hydrogel formation. Becomes like a big Gummibärchen. I don't know the English term. So like a rubber thing, but yeah, it can be effectively purified. Yes.
Awesome. Another quick question then. Can the PEC reagents be supplied with BSE TSE certification if that's necessary for the peptide quality requirements?
Yes, that's no problem. We have done this already for several customers. So yeah, the material has no animal origin, so we have the certificate already prepared. Sorry.
Very nice. And here's another question from one of our viewers. If the peptide has an N-terminal proline, does the PEC linker still react as effectively as opposed to if the peptide has a free, primary amine as the N-terminus?
Yeah, that's no problem. I mean, with the especially with the high throughput kit, as I told you, we are also currently providing peptide libraries for potential customers, and for that we are purifying on a regular basis, like all 20 amino acids on the N-terminus. So, and we have not seen any differences in coupling efficiency. So proline for sure is no problem. For some reason, like it sometimes seemed that threonine doesn't give as good coupling efficiencies, but I think that's not true. We had a deeper look in this, and this was just a rumor, basically.
This is an interesting question. Can the PEC technology be adapted for peptides with N-terminal modifications, like acetyl, like if you have an N-terminal acetylation or a dye? So I imagine that you'd want to install the linker on a lysine side chain if that was available in the sequence. Is that possible?
Yeah, exactly. I mean, this is the only limitation the technology has, that on the N-terminus is the linker, and or in the standard form. And if you want to have something on the N-terminus, you have to go over the side chains. You can do it by a lysine. This is the easiest, and we have done it, and also customers have done it several times, especially with, I know that one academia group in Portland has done it effectively, that they introduced as a last residue a lysine, and had the PEC linker on the side chain, and had on the N-terminus the fluorophore. So fluorescent dye. Yeah, this is feasible.
And is there a minimum length that you've found in order to be able to use the PEC technology effectively? For instance, can it be used to purify tetramers or pentamers?
A minimum length?
Yes.
Yeah, I just would not recommend to use the technology for those, because I think there you can more effectively use other purification methods, but there is no minimum. Actually, we found that that small peptides, like the minimum we purified, once we did a trimer actually, and we also did many 6 mers. And the problem there is, if you use our standard conditions, it involves a final ether precipitation actually, when you don't use a high throughput version, but the normal research kit format. And we found that with linker on even the small peptides, they precipitate quite well. in ether, but the small ones without linker don't precipitate as well. So we got big losses in peptides. And maybe to add on this, the technology makes way more sense for long peptides, because as you can imagine, you always have, even after improvement of the SPPS, you still have some couplings that are not 99.999% in coupling efficiency. So you always, after each coupling, you build up truncations. So we have case study on our website where we have purified an 84-mer PTH sequence. And there you really see that you have like a triangle shape of the desired peptide with lots of truncations underneath. And we can selectively like fish out this one. So it makes way more sense for long peptides.
And on the flip side to that question is, what is the longest peptide that can be purified using PEC? What is the long? You mentioned PTH 1 through 84. Is that the longest you've been able to do, or have you achieved longer purifications?
That's the longest we did.
Okay.
Maybe this adds on a little bit to the first question, or one of the first was a proline. So there's no limitation in amino acids, but we see that as in all couplings, the efficiency of linker coupling may go down when there's aggregation on the SPPS resin. So, but you can couple the linker also by using microwave or by heat. So there is no length border, but the maximum we have tested is 84 residues.
Right, and regarding protecting groups, if the peptide has alloc or benzyl protection on the side chains, which are usually deprotected using palladium, can that same palladium catalyzed hydrogenolysis also reduce the azide to the amine and trigger the release of the peptide?
We have not tested that. So we have the first linker system was basically cleavable. I can say for sure that the alloc deprotection does not harm the basic linker, because there I also synthesized the lyroclatide and did an alloc deprotection of the lysine, and then I added the palm glue building block. Therefore, I can say that on SPPS polystyrene resin, the coupling of this particular palm glue building block also doesn't work well, or it's not so easy. Let's put it this way, at least in my hands. But from the reductively cleavable linker, I think it won't be reduced, because what we I'm pretty sure it will work, because it's a heterogeneous catalysis. So we have actually tested many things when we wanted to find a good reducing agent for the linker. Also, we tested nitro linkers, and we found that like all catalysts we used, most of the catalysts eventually work heterogeneously. So it did not work to reduce the linker. So I'm pretty sure it would work, but it has to be tested. Yeah.
And to follow up on the question earlier regarding BSE TSC statements, we have a question asking, is this technology GMP ready, and have you filed a DMF for the resin production as a medical device?
Sorry, could you please repeat DMF
Sure.
as a medical?
Sure. So this viewer is asking if the peptide Easy Clean technology is GMP ready, and if you filed a DMF or a drug master file for the resin production.
No, we have not yet. Actually, we have talked to Bachem about this. What is necessary, and the current belief is, and this is currently under investigation, that there, I mean, for the API, a drug master file has to be filed, yes, but that the linker and the filter material doesn't have to be produced under GMP conditions because it's not in the final product. It's basically can be regarded as the SPPS resin. And of course, it has to fulfill highest quality standards. And we as a manufacturer, we have to be audited and all that. But we don't have to file a drug master file for the linker and the filter material.
We have two questions asking for recommended conditions for oxidation of, for reduction and oxidation to form disulfide bonds using this process. Is this published somewhere?
Not yet. So we quite soon will bring out a case study on our homepage. This is the case studies we have out there. That's just like a two-page white paper that will give some details on that, also on the two examples I've shown here. What we found, what works good is we have our standard DTT linker reduction. This also reduces potential disulfides that may have been unwanted and be there before. And then the oxidation gave the best results when we used this, whether Rabenstein reagent or NCS, so N-chlorosuccinimide. But depends also from peptide to peptide, and you would have to have a look on each peptide. But if you want to have a like one size fits all solution, then this Rabenstein and NCS was the best in our hands.
Thank you. And so these are the last two questions for this session. So the next question is, what's the highest purity that you have achieved for modified peptides using the PEC technology?
For modified peptides, I'm not 100% sure, but I know that so when you do this HPC pre-purification, and then you modify your peptide and you cleave off, you can achieve very high purities of 95 or 96% purity. But in the general PEG process, we also achieved 100% by our UPLC integration. So I know 100% is not really there, but at least according to our analytics, it was 100%.
Very impressive. And final question is, does the PEC technology tolerate modifications that may be done via free radical chemistry?
I don't know. It may cleave the linker. I'm not sure. I just know that from my PhD work, where in the group of Professor Oliver Seitz, there was also radical chemistry of linkers, and this goes crazy ways. So it's really hard to foresee, I would say. This would have to be tested.
All right. Thank you so much, Robert, for answering all of the viewers' questions. And thank you again to all our viewers for submitting such interesting questions. I'd like to thank everyone for their attendance. And if we weren't able to get to your question, I do apologize. And if you really would like an answer, please send an email to webinar@bachem.com. And I promise you, we will indeed, check that inbox and read those emails and get back to you with the answer to those questions. In the meantime, you can always follow us on social media as shown in the icons below. And with that, I'd like to thank Robert once again for this interesting presentation. And again, to all of our viewers for taking the time to watch this. Thank you very much.
Yeah, thank you also very much for your attendance, and Bachem for giving me the opportunity to speak here.